Ebola virus and vaccine. Ebola virus, EBOV, is a filovirus that causes Ebola Virus Disease, EVD, a severe and often fatal hemorrhagic fever in humans and non-human primates. It is one of the most lethal viruses known, with case fatality rates ranging from 25% to 90%. Origin and history. Discovered in 1976 during simultaneous outbreaks in Anzara, Sudan, and Yambuku, near the Ebola River, Democratic Republic of Congo. The virus was named after the Ebola River. Reservoir host, believed to be fruit bats, Pteropodidae family. Transmission to humans, through contact with infected wild animals, bats, monkeys, or their body fluids. Human-to-human transmission. Direct contact with blood, secretions, organs, or body fluids of infected individuals. Contact with contaminated surfaces or materials. Via funeral practices involving the deceased. Major outbreaks. Year. Region. Impact. 1976. Zaire, DRC, and Sudan. First known outbreaks. 1995. Kikwit, DRC. Greater than 250 deaths. 2014, 2016. West Africa, Guinea, Liberia, Sierra Leone. Largest outbreak, 28,000 cases, greater than 11,000 deaths. 2018, 2020. Democratic Republic of the Congo, DRC. Several regional outbreaks. 2021, 2022. Guinea and Uganda. Re-emergence due to viral persistence. Structure of Ebola virus. Feature. Description. Family. Phylogenetic. Genus. Ebola virus. Type. Enveloped, filamentous virus, unique thread-like shape. Genome. Single-stranded, negative-sense RNA, 19 kb. Virion length. 800 to 1000 nanometers, can reach up to 1400 nanometers. Envelope. Lipid membrane derived from host cell. Proteins. 7 main structural proteins. Major structural proteins. Protein. Full name. Function. GP. Glycoprotein. Mediates attachment and entry into host cells, major surface antigen. NP. Nucleoprotein. Encapsulates viral RNA. VP24. Viral protein 24. Structural integrity, interferon inhibition. VP30. Viral protein 30. Transcription activation. VP35. Viral protein 35. Interferon antagonist, cofactor for polymerase. VP40. Viral protein 40. Matrix protein, assembly and budding. L. RNA-dependent RNA polymerase. Viral genome replication. Pathogenesis, how Ebola causes disease. 1. Viral entry. The virus enters through mucous membranes, wounds, or contact with infected fluids. The GP, glycoprotein, binds to host receptors, example DC sign, TIM1, NPC1, on macrophages, dendritic cells, and endothelial cells. Entry occurs via macropinocytosis and endosomal fusion. 2. Replication. Once inside, the viral RNA is released into the cytoplasm. L polymerase transcribes viral mRNA and replicates the genome. New virions assemble and bud from the host cell membrane. 3. Immune evasion. VP35 and VP24 inhibit interferon, IFN, responses. The virus disables antiviral signaling, allowing unchecked replication. 4. Systemic spread. Infects macrophages, dendritic cells, liver, and endothelial cells. Damages vascular endothelium, leading to vascular leakage, internal bleeding, and multi-organ failure. 5. Cytokine storm. Overactivation of immune cells releases massive cytokines, rightward arrow fever, shock, and tissue damage. Clinical manifestations. Stage. Symptoms. Early, 2 to 21 days. Fever, headache, muscle pain, sore throat. Progressive. Vomiting, diarrhea, rash, impaired kidney/liver function. Severe. Hemorrhage, multi-organ failure, shock, death. Why Ebola is an emerging disease. Zoonotic spillover, from fruit bats to humans through wildlife contact. Human-to-human transmission, spreads rapidly in areas with weak healthcare systems. Viral persistence, virus can survive in immune-privileged sites, eyes, semen, placenta, re-emergence after recovery. Global travel, can spread across borders quickly. Lack of medical infrastructure, limited access to early diagnosis and supportive care. Mutation potential, RNA virus with high mutation rate, challenges vaccine development. Need for an Ebola vaccine. High fatality rate, up to 90%. Recurrent outbreaks in Africa and risk of global spread. Protect health workers, close contacts, and endemic communities. Control epidemic cycles and prevent future pandemics. Provide long-term immunity in populations at risk. Antigenic components used for Ebola vaccine development. The most immunogenic components, used as antigens in vaccine design, are viral component, function/immune role, vaccine use, GP, glycoprotein, major surface antigen, mediate cell entry, induces neutralizing antibodies, main antigen in almost all Ebola vaccines, VP40, matrix protein, helps in viral budding, induces cellular immune response, used in multi-component or VLP vaccines, NP, nucleoprotein, induces CD8 T cell responses, enhances cell-mediated immunity, VP24 and VP35, interferon inhibitors, contribute to immune evasion, targeted for T-cell-based vaccine designs, vaccine design strategies against Ebola virus, 1 viral vector vaccines, use harmless viruses, like adenovirus or vesicular stomatitis virus, to deliver Ebola genes. Example. Description. Status. rVSV-ZEBOV, ERBO registered. Recombinant vesicular stomatitis virus expressing Ebola GP. Approved by WHO and FDA. Ad 26.ZEBOV/MVA-BN-Filo. 2-dose vaccine, Johnson & Johnson, using adenovirus plus MVA vectors. Approved, EU, Africa. 2 DNA vaccines. Plasmids encoding Ebola GP or NP are injected rightward arrow host cells produce proteins rightward arrow immune response. Safe and stable but may require boosters or electroporation. Examples. NIAID GP-based DNA vaccine. Multi-epitope GP plus NP constructs. 3 subunit vaccines. Contain purified recombinant GP or VLPs, virus-like particles, mimicking Ebola structure. Induce strong antibody and T-cell responses. Examples. GP-based nanoparticle vaccines. GP plus VP40 VLP vaccines, preclinical. 4 mRNA vaccines. Encode Ebola GP or GP plus NP, leading to rapid immune activation. Quick to design and modify for new variants. Example, Moderna mRNA 1325 candidate, preclinical trials. 5 peptide/multi-epitope vaccines. Contain selected T-cell epitopes from GP, NP, and VP24. Designed using bioinformatics and immunoinformatics tools. Mechanism of immunity from Ebola vaccines. GP antigen, induces neutralizing antibodies rightward arrow prevents viral entry. NP/VP40 antigens, stimulate CD8 cytotoxic T cells rightward arrow kill infected cells. Combined humoral plus cellular immunity gives long-lasting protection. Ideal Ebola vaccine antigen should induce strong neutralizing antibody response. Elicit robust cellular, T-cell, immunity. Be conserved across Ebola strains. Be highly immunogenic but non-pathogenic. Mimic natural viral structure. Be suitable for different vaccine platforms. Be stable, scalable, and safe for global use. Avoid immune evasion and antibody-dependent enhancement, AID. Challenges in vaccine development. Need for protection against multiple Ebola species, Zaire, Sudan, Bundibugyo, etc. Maintaining vaccine stability in tropical conditions. Ensuring safe use in pregnant women and children. Avoiding immune overactivation or side effects. Ebola virus remains a global health threat due to its high fatality rate, zoonotic nature, and outbreak recurrence. Vaccine development focusing on the GP antigen has already saved thousands of lives. Future vaccines aim for broad protection across multiple Ebola species using multi-antigen, mRNA, and nanoparticle-based platforms. Thanks for watching.